Many thanks to the OIS team, and I'm really glad I got a walk-up song. So I'm a big baseball fan, so I guess that's my first forward-looking statement. I think the Dodgers are going to win again next year. So you can find the rest of our forward-looking statements. That's not going to be in the Qs and the Ks, but what will be is that Kiora is a clinical stage retina-focused company. If you took a look at the Polaroid of Kiora today, we have two assets in development, 301 and 104. One is a photo switch focused in rod cone dystrophies, and 104 is targeting retinal inflammation. We are proud to have partnered 301 early in the year with TAEA. The terms of that deal you can see on the slide. And 104 is something we still retain full commercial rights on. It has been a very difficult environment. Certainly, we heard a little bit about that yesterday for microcaps such as us. We are fortunate in the sense that as of September 30th, we do have $30 million plus in cash, and that funds operations through and into 2027. So doesn't remain or make us complacent, far from it. We remain incredibly diligent about what we do, and we are a lean organization. So if you look at our pictorial view of our pipeline, those of you who do know us, you may know us mostly as a company that's focused on, like I said, the rod cone dystrophy and our photo switch, and we do have our 104 program in retinal inflammation. And so I'm going to switch it up a little bit and talk first about 104. We do see a need for the continued development of non-steroidals to treat retinal inflammation. I mean, retinal inflammation, we all know underlies so much of the disease. You got to drive through that neighborhood ultimately to get to a disease. And that having in the arsenal a non-steroidal, in this case small molecule approach, is something that we believe absolutely has room to innovate and has room in the arsenal, frankly, of either the general ophthalmologist or the retina specialist. I'm sort of reminded of the fact that if you look at rheumatologists back in the 80s, they had methotrexate and steroids, and if you look at their arsenal today, it literally is populated with probably 25 to 30 biologics that they have now the ability to treat their patients with. There's no reason why this community still can't get to that same type of approach when we think about disease. So certainly looking at macular edema, that would be one of the certain indications, or at least conditions in which inflammation plays a role. And specifically 104 is a member of a validated class of molecules known as DHODH inhibitors. So DHODH inhibitors are well established. They're disease-modifying agents, today on label for RA, MS. And what they do is they are an enzyme, or they're a small molecule that inhibits the DHODH enzyme, which effectively starves cells of their de novo pyrimidine synthesis. So if you're trying to mount an immune response and you don't have a pool of pyrimidines available, you're going to be able to really clamp down on that immune response, specifically Th1 and Th17 cells, as well as get a downregulation in a host of cellular messengers that are associated with those cells. So the clinical aim is then and in any condition in which you have inflammation, we've taken this same class of DHODH inhibitors, which is really a third generation version of that, put it in an IVT format, formulated as such, and then looking at a variety of indications where retinal inflammation will play a role. Our first-in-man proof of concept study, which was published about 18 months ago, this was a single-dose administration in patients with posterior non-infectious uveitis. We found a 3-line improvement, just giving you some reference points relative to steroids and the biologic that is available on market. This was, again, ascending dose administration of the drug. We also saw 40% of the eyes in that study that had vision-threatening macular edema or cystoid macular edema at baseline had a reduction, a meaningful reduction, and that correlated also with a CST. That is leading to Clarity, which is our phase 2 study, which we will kick off in Q1 of next year and read out in Q2 of 2026, where we're going to take 8 patients with macular edema that have a variety of different underlying pathologies look at a low dose, look at a high dose, then decide just based on safety and do an expansion cohort where we take one of those doses and randomize them either to a biweekly or a monthly administration. And then we'll read out those patients, looking obviously at safety tolerability, but standard right down the fairway clinical endpoints of BCVA, CST, and patient-reported outcomes. So switching gears, given the time, to 301. 301 is a molecular photoswitch. This is a very innovative molecule that changes shape in response to light. Patients that have degenerated photoreceptors, well, let me take a step back. This is really optogenetics without the genetics. So it is a small molecule trying to target or restore vision regardless of the mutation or the underlying mutation. And the mechanism by which it works is it targets retinal ganglion cells and makes retinal ganglion cells photoreceptive. And it does so by using a small molecule that gains access to the retinal ganglion cell that sits downstream from degenerated photoreceptors. And in the presence of light, these molecules change shape and result in depolarization or activation of the retinal ganglion cell, which then gives rise to the signal to the optic nerve. In Abacus, I'll skip we presented this original data set at the 2023 AAO. There was a first-in-man study, single-dose administration of 301. It was safe and well-tolerated. All the adverse events were related really to the injection. This is, as I mentioned, a single, an IVT administration. When you look at the improvements in vision, we're really looking as a first-in-man study, is there any non-random signal that we can see as a non-controlled first-in-man study? When we looked at kinetic visual field, we found about a 25-degree improvement in visual field. We saw a reduction or an improvement in visual acuity with a reduction in the logMAR. And then, of course, putting patients through functional testing, we saw an improvement in this case, an example of that in a maze-like testing. So when you look at the full complement of vision, you saw a lot of correlation between the different measures of vision that we looked at, and ultimately that led, in addition, we looked at functional MR to could we see the brain lighting up, and we in fact did. We saw statistically significant improvement, and you can see the waning or the improvement at day 2 and then the waning at day 29, and that is consistent very much with a small molecule approach to vision restoration. That leads to Abacus 2, which is a study. Main differences between Abacus 1 and Abacus 2 are going to be, this is going to be a 36-subject randomized masked study where we are then not only treating bilaterally, we'll of course be using a control, and we will be using and doing multiple doses in these subjects. This is slated to kick off also in Q1 of next year and read out in 2026. So thank you very much for your time. I appreciate it, and have a great rest of the day.